welcome to this X-Planner. Okay, I am absolutely thrilled to guide you through today's topic because, honestly, we're looking at what might be the single most fascinating medical paradox of the last 50 years. We are going to explore how a 1960s battlefield anesthetic, something known for producing these wild out-of-body experiences, completely revolutionized our understanding of severe depression. It's a story of accidental discovery, really complex brain chemistry, and a fiercely debated scientific mystery.
Okay, let's dive right into this. To understand why the medical community is paying so much attention to this, you really just need to look at this number right here, 71. In a landmark 2006 study, researchers found an absolutely astonishing 71% response rate in patients with treatment-resistant depression. And here's the truly incredible part. That response happened within just 24 hours from a single dose. I mean, think about that for a second. Traditional SSRI antidepressants can take weeks or even months to start working, if they even work at all for these patients. This molecule flips the script entirely. It offers profound relief in literally a matter of hours.
So how exactly are we going to unpack this today? Well, here is our roadmap. First, 1. An accidental antidepressant. Then, 2. The glutamate paradox. 3. Rebuilding the brain. 4. The dissociation debate. And finally, 5. The real risks. Let's get right into it.
All right, Section 1, an accidental antidepressant.
So this is the origin story of how a chemical designed to fix a problem drug's side effects somehow became a psychiatric breakthrough. Our timeline kicks off back in 1962. A chemist named Calvin Stevens synthesized ketamine as a gentler, shorter-acting alternative to PCP, which was causing severe agitation in patients at the time. By 1964, human trials were underway. When anesthesiologist Edward Domino administered it, volunteers reported feeling utterly detached from their own bodies. In fact, Domino's wife brilliantly coined the term dissociative to describe it. Because it remarkably preserved breathing and blood pressure, the FDA approved it in 1970, making it a staple in operating rooms and on battlefields. But the real plot twist, that happened in the year 2000. Researchers Berman and Crystal at Yale gave a really low dose to a small group of severely depressed patients, and those patients felt better within hours. Fast forward to 2019, and a variant of this molecule, S-ketamine, marketed as Spravato, became the first genuinely new mechanism antidepressant approved by the FDA since Prozac.
Now, what makes this application so unique is really all about the dosing. The clinical antidepressant dose is about 0.5 milligrams per kilogram, infused slowly over 40 minutes. That's what we call a sub-anesthetic dose. The dose required to actually put someone under for surgery and erase surgical pain is roughly 10 times higher. It's the exact same molecule, but at a fraction of the dose. Instead of shutting the brain down for surgery, it somehow lifts the crushing weight of severe depression.
And that brings us to Section 2, the glutamate paradox. This is where we get into the central scientific puzzle of how this drug actually interacts with our neural circuitry. Now, what's really interesting here is how ketamine distinguishes itself from classic psychedelics. You might hear them grouped together in the media, but neurologically speaking, they are entirely different beasts. Classic psychedelics like psilocybin or LSD work by activating the serotonin 2A receptor. Ketamine, on the other hand, doesn't act on serotonin at all. It's a non-competitive, open-channel blocker that targets the NMDA receptor, which is the main excitatory switch in the brain's glutamate system. That specific action is exactly why it's classified as a dissociative rather than a psychedelic. Let's move to and see how this builds into what scientists actually call the disinhibition hypothesis. Because if you're blocking the brain's main excitatory receptor, you'd naturally expect brain activity to slow down, right? Well, paradoxically, the exact opposite happens. Step 1. Ketamine preferentially blocks the NMDA receptor sitting on fast-spiking GABA interneurons. Think of these GABA neurons as the brakes on your brain's engine. Step 2. By blocking them, those inhibitory brakes are effectively silenced. Step 3. Without those brakes, the main output cells, the pyramidal neurons, are disinhibited and they just start firing freely. Step 4. This unleashed firing results in a sudden, massive surge of glutamate that hits a completely different set of receptors called amphoreceptors. And it's this transient burst of signaling that sets the entire healing process in motion.
All right, Section 3, rebuilding the brain.
So what happens after that burst? Well, the initial glutamate surge isn't actually the cure itself. It's really just the spark that triggers a remarkable downstream physical transformation. To understand this, we need to introduce a relatively new term in neuroscience, psychoplastogen. This refers to a substance that rapidly promotes neuroplasticity. It literally alters the physical structure and function of the brain.
You see, chronic stress and severe depression are deeply associated with the physical withering and loss of synapses in the prefrontal cortex. A psychoplastogen acts basically like fertilizer, rapidly regrowing those lost connections, which appears to be what sustains the remission of depressive symptoms long after the drug has completely left the patient's system.
And this brilliantly illustrates exactly how that physical regrowth happens at the cellular level. Following that glutamate surge we just discussed, two major cellular pathways are rapidly activated: the MTR pathway and BDNF, which is the brain's key growth factor. A landmark 2010 science paper from Ronald Duman's lab showed that activating these pathways grows brand new dendritic spines. Those are the physical connection points between neurons within mere hours. Now, as a quick caveat, we absolutely have to note that this vivid synaptic evidence primarily comes from animal models like rats. We infer this mechanism is happening in humans, but, we can't directly view microscopic spine growth in a living human brain just yet.
Moving on to Section 4, the dissociation debate. As promising as all of this sounds, the clinical community is actually highly divided over one specific aspect. This is literally the million-dollar question the whole psychiatric field is circling right now. Is the trip the medicine? We know that at clinical doses, ketamine reliably produces this floating, detached feeling, a dissociation from the body and the self. Some early analyses found a direct correlation here. The more intense the dissociation a patient experienced, the better their antidepressant response seemed to be. That strongly hinted that the subjective, conscious experience of the drug was fundamentally intertwined with the healing process.
So how do you test that hypothesis? Well, an incredibly 2023 trial by Lee and colleagues did exactly that. They took depressed patients who were already going in for routine surgery and gave them either ketamine or a placebo while they were completely unconscious under general anesthesia. Because they were asleep, they couldn't consciously experience any kind of trip. The results? The ketamine group showed no better antidepressant response than the placebo group. While this trial was small, it objectively suggests that the expectation of getting a powerful drug, combined with the conscious experience of the dissociation, might be driving a lot of the actual benefit. It really highlights the active placebo problem in previous trials, leaving the true mechanism of action genuinely and hotly debated.
Which leads us to Section 5, the real risks.
Now, it is absolutely vital that we separate the controlled clinical triumphs from the severe dangers of unmonitored use. While a monitored clinical infusion has relatively low physiological toxicity, chronic recreational use is a completely different story. Heavy, repeated use can lead to something called ketamine cystitis, which is a severe and sometimes irreversible disease that damages the bladder. Furthermore, heavy users show measurable cognitive deficits over time. Additionally, the drug carries a very real risk of psychological dependence. It is a Schedule III controlled substance for a reason. There is an enormous critical gulf between receiving a precise 0.5 milligram per kilogram dose monitored by a doctor and unmonitored high-dose recreational binging.
The gravity of these risks was recently, and tragically, underscored by the widely reported death of actor Matthew Perry. His passing, which was ruled an accident from the acute effects of the drug, objectively highlights the extreme danger of utilizing this substance outside of strict, rigorous clinical supervision. This explainer is educational, but it's just so crucial to respect how dangerous this tool can become when the clinical guardrails are completely removed.
Which leaves us right back here at the profound medical mystery we started with. Ketamine basically broke every rule in psychiatry. It treats despair through a forgotten receptor, it works in hours instead of weeks, and it shifts our whole view of depression from a chemical imbalance to a disease of lost neural connections. But as we look to the future of this research, we're left with this final, really provocative thought. When a drug both physically rewires the brain and psychologically dissolves the self, which one is really doing the healing? Are we fixing the hardware or are we rebooting the software? The answer is still out there, waiting to be fully discovered. Thank you so much for joining me on this explainer. Keep asking questions, and I'll catch you next time.